All right. Hello, everybody. I hope you're having a good day, morning, evening, noon, wherever you are. Today, our talk's going to be about machine learning and neuroscience, a topic which I'm very excited about as well. The lead will be Robert Boshra, and I'll just quickly introduce him before passing it on to the speaker. So Robert Boshra is an active researcher in neurophysiology and the application of machine learning in the clinical setting to improve assessment and outcomes. He's worked in brain injury target objective concussion assessment using EEG and ERT. His work also investigated automated detection of consciousness in coma and vegetative states. He has interdisciplinary training as a computer scientist, neuroscientist, and biomedical engineer. Robert is an alumnus of McMaster's AI-focused McData Institute and an active affiliate of Toronto's world-renowned Vector Institute of AI. Robert is the director of AI and technology at Vox Neuro and is currently leading a project with funding awarded by the National Research Council to scale the company's services across Canada. So, very excited to hear from Robert, and I will pass it on to you, Robert.
Thank you very much, Susan, for the introduction. I'm very excited to be here and very excited to kick off this new stream. So I prepared these slides and this talk just to give a bit of a very broad context of what can be done by machine learning and neuroscience, and I think this only scratches the surface of what's going to be discussed in the next few months. So just before we get going, I just want to say that some of the work that I will be talking to here has been done in collaboration with a phenomenal group of individuals at McMaster and other places as well, and that the work that has been done by me and my entities on the right there, and just a disclaimer that what I'll be discussing is my opinion and doesn't really represent the views of any of these entities. So just to start things off, I just want to set the stage by, I know everybody knows the brain, everybody knows what it does nowadays. It wasn't known as the center of consciousness and cognitive function before, but now we know that. And despite that knowledge and that development of our understanding of the brain and neuroscience in general, our understanding still lacks in a lot of different aspects, and which is very exciting to now see the development of machine learning and how the interaction between them has really pushed things further a lot more. So to dive a little bit into the low level of the brain, so as most would know, the brain is formed by all these neurons forming this intertwined network of different cells that communicate with each other using electrical signals back and forth, and that there are postsynaptic potentials that actually using different neurotransmitters, they can actually send signals between cells as well. And it's not the single cell, although the single cell is very complicated and complex. It's the network of these cells that actually form all these very fascinating things that the brain can do, from cognitive function to creativity. So just to set the stage of how to actually study neuroscience, just a very toy model of what can be done. If you think of the brain as a black box of sorts, and you want to understand what is happening in it, you don't really care about the single cells, but how does it actually work? So you take a bunch of inputs, or you can take one of the inputs, so it could be stimulation, like auditory, visual, like video, an image. You could take pharmacological, so you can apply anesthetics, or another kind of medicine that passes the blood-brain barrier. Or you can just tell the person with the brain, the owner of the brain, to do a particular mental task, like memorize a particular thing, or count from 1 to 10. And you provide that input to the brain, and then you look at the output. And that output could be behavioral, just like in very simple terms, like what will they say? And in a more of an autonomic aspect, like if you put an EKG on their heart, does their brain rate go up? The heart rate go up? Are they affected by what you've just stimulated them with? Okay, so this is kind of the paradigm, and without looking into the what has appeared in the last 100 years or so. So enter all these different ways to actually look at the brain, either functionally, anatomically, or neurobiologically. So you can look at electrical signal of entire neuron populations and see what they are doing, negative, positive, and over time. You can look at the magnetic field of that electrical field created by the neuron populations, or you could look at the blood oxygenation level of different brain regions and characterize them in space using magnetic resonance imaging. Okay, so this is just functional, but then you can also look at the anatomy. So you can look at the MRIs or the diffuse tensor imaging of the brain and look at, well, if you apply some stimulation over time, how does the brain's shape change as well? And after expiry of the person, which actually has happened in some areas, you then study the neurobiology and actually after in vitro. So all of this is very complicated because with each of these different things, you can target a completely different question. But then you add the fact that different brains are vastly different, and that the brain in health is very different from brain in pathology, and this just, the combinatorics of this just blows everything out of the water. And with pathology here, I mean brain injury, Alzheimer's, Parkinson's, anything that affects the brain, really, and mental illness. So reeling this back into the realm of, okay, so why machine learning in this case? And let's just get into that. We have the brain. We know that the brain is responsible for perception and intelligence, and we really want to emulate it in what is today in the computer. And this is kind of where things started. It's the brain inspires the different models. So you started with single perceptrons and Hopfield nets, which I don't even think you hear about anymore. And all this started this spiral. Of course, it went up and down, but now it's very prominent, like artificial neural networks and deep learning. It's a big thing. And then you take it back, and this is kind of what I really want to bring here, of using these tools to try to funnel it back and answer those biological questions that we have. And I've heard this in a podcast, and it was Dr. Carl Friston have put it very well, saying that this loop between inspiration by biology, analyzing biology, and this loop is what is going to really revolutionize both fields, actually. But for the purpose of this stream and for the purposes of this talk, I'm really going to focus on the analyze piece, and I'll promise that I won't go into tangents on the inspiration piece at the top there. So just to summarize, just different targets in neuroscience that I very much look forward to discussing in this stream. It's going to target consciousness, and not just the philosophical aspects, which are also very interesting, but just how can we investigate it. Language, which is actually quite prominent in machine learning and neuroscience nowadays. Pathology, just different things that can affect the brain. Sensory processing, like, okay, so you see something, how does your brain actually react, and what lights up and what doesn't, and how can we leverage that into how can we leverage machine learning into understanding that more? And lastly, which is very complicated and it's very loaded, those 2 or 3 words, just cognitive processing and memory, executive function, and all these intricate parts of what makes us human. So for the purposes of this talk, I want to discuss 3 different papers that kind of give a very broad overview of what can be done. So the first is modeling of neurogenesis in the hippocampus, a structure in the brain. Probing disorders of consciousness. So after coma, do we know that that person has emerged or not using brain-computer interfaces? And lastly, just the effects of traumatic brain injury on brain function, and how can we tell whether somebody has been affected or not. So I'll start with this. It's a paper published in 2015 titled Neurogenesis Paradoxically Decreases Both Pattern Separation and Memory Interference. So this paper holds a sweet spot in my head, because this paper, so Rory, the first author, actually started his master's with me, as we were both starting neuroscience at the same time, and he also has a background in computer science, and it kind of started this whole thing in my head of like, the machine learning can be used in such interesting ways in neuroscience, and it started with using restricted Boltzmann machines, and it kind of spiraled from there. So it was very exciting seeing this come to shape, basically. And to start, I just want to give a very broad overview on the hippocampus. So the hippocampus is a deep structure in the brain. If you drill deep down temporally, don't do that, and you will find this shape over there in red. So that the hippocampus has been very heavily studied recently, because you can really link it to a lot of things that we don't understand, and we try to emulate, but we can't really, like memory and learning. And when you lesion that area, you start to find that people cannot form new memories or can't learn new skills. It's very interesting. The problem about that is, as opposed to more cortical functions or cortical structures, this is very deep down. And when you drill that deep down, the things like EEG, electroencephalography, or magnetoencephalography, which work a lot more on the cortex because they don't go inside the skull, they cannot see what's happening in the hippocampus, which gets you to think and invest a lot more in pure theoretical modeling of the brain and this structure of the brain. So the hippocampus is also very, very interesting in the fact that it conveys neurogenesis. So I think not even that long ago, people thought of the brain as this, it's one and done. It develops, and then this is how it is going to be for the rest of your life. It's actually going to deteriorate, and then you die. But in more recent work, it's been understood that actually there are new neurons, there are new cells that start to take shape in the hippocampus. But nobody really knows the purpose of it. They know that if neurogenesis is affected, there are certain problems. But what exactly? And this study is basically looking at that and trying to model those new neurons and new cells using restricted Boltzmann machines. So restricted Boltzmann machines, to anybody not familiar with them, they were quite famous and quite well utilized. Now I think they've fallen out of favor a little bit, just because you can skip it and go directly into deep learning. But basically, in very broad terms, you have an input, you have an input layer and one single hidden layer. And of course, you try to maximize, you try to learn the weights connecting between them. There are no interactions between one layer. And it's generative and it's stochastic. So typically, you learn binary, so binary inputs, binary outputs. So in this particular study, the way it was done is that the input was treated as the entorhinal cortex, which is again cortical structure, and then the hidden layer was treated as the hippocampus. And during different experiments and different setups, what the authors did was add new items to the hidden layer, which is equivalent to neurogenesis, and apply a a constraint of sparsity on the hidden layer as well, because the hippocampus tends to be rather sparse. And then they just examined like how much does the neurogenesis get affected by that neurogenesis and by that sparsity constraint. And I would really be underselling it if I'm saying this is it, because this is such a minefield of different things that happen in the hippocampus. But very broadly, the authors concluded that neurogenesis is very much important in pattern separation, and that those new neurons that are hyper excitable are very important to learn the new patterns and reduce the interference between them. However, it actually decreased pattern separation, which is kind of counterintuitive, because you would think that new neurons would enable you to learn a bit more. But that's when the model might be now hinting at biology, because we're not perfect. We don't perfectly separate between patterns, and this is where the future work of this paper is going. So again, very, very interesting, and just the dynamic of the maturation of the young neurons, because the young neurons don't stay hyperexcitable for the rest of the lifespan. They mature, and in doing so, the reduction of interference is better and better. So,
all right, Robert, we have two questions actually.
Great.
So the first one is, could you quickly go through what pattern separation means in this context again?
For sure. So going back to this, let's say the input layer has, so they simulated different patterns, let's say like 1011, okay? And you try to add a bit of noise. So let's say it's 1101 and you remove the 1 and just have that be noise. How does that model, that learned model, separate that from if the binary input was completely different? So let's say 0001. So it's a completely different pattern plus a bit of noise. And how good is the model at separating those two noisy inputs? And is it actually able to identify and separate them?
I guess noisy here in the terms of, I guess, like the neurology context, like what might that noise kind of be? Like signals or like...
Mm-hmm. So noise in the context of the model itself is just different randomly associated one of the bits, right? Because the input is binary, so you just flip one of them. So it's a noisy input. It's like noisy autoencoders.
Okay.
Right, but in the context of biology, that is different, because our sensors as humans and the sensors that get into the brain are very noisy. What you see is not what's actually happening. So this is kind of the equivalent of that, the model versus the biology.
Gotcha. So there are some, I guess, biologically there are some noises that, I guess, like noisy signals.
Mm-hmm. Biology is very messy.
Yeah. Gotcha. That's true. The second question is, what is is there a theoretical, I guess, background or justification in using deep learning to emulate brains?
For using deep learning to emulate the brain?
Mm-hmm.
I mean, it actually goes the other way around, right? Because the brain was the source of deep learning. It's what started this entire thing of, okay, well, the brain looks like this. It's because the brain looks like a deep network, right? So in order to try to understand the brain, you need something that looks like it, and try to set the inputs and outputs to something that you would provide the brain, and see and actually investigate those layers and see if those layers shed any light on what could be happening in the brain. There is one thing that I just want to point out, is that in deep learning you typically have a set number of neurons, right? And those neurons are feed forward. There's back propagation but in real life in biology, back propagation, people don't really know if that is the case in biology. Neurons are very different. Different neurons do different things. So it's definitely an abstraction of the brain.
Gotcha. Thank you.
Yep. Okay. Well, if there are no more questions, I'll move ahead to the second paper, which is by Lule, I believe, and this is a group led by Stephen Loris, I believe in Belgium. Again, very big group that is great in the context of consciousness, and they've been doing this for a very long time with amazing output. I chose this particular paper because it ties in with brain-computer interfaces, so which is more clear to discuss, and it's also it has a very easy read-through. So right before we jump into this, I just want to bring you up to speed on what the concept of evoked potentials is. So the brain, again, generates electrical signals coming out of populations of neurons, and if you stimulate the brain by a particular sound, let's say a tone, and that tone happens, let's say 1,000 times, and you take the brain response right after that sound, the signal coming out of the brain isn't just noise anymore, or isn't you can't really tell what's happening. It actually starts to form these little peaks and troughs in what you receive. This is after averaging, because you increase the signal to noise ratio, and this is what is called the evoked or event-related potential. So nowadays, it's not just one electrode, it's a bunch of electrodes, and if you stimulate the brain in particular ways, you receive a different set of these shapes and responses. So time here is x-axis, amplitude is y-axis. So again, very complicated. I just want to point your attention to this P300, because literally each one of these is a different field per se, if you really want to get down to it. But the P300 is very robust, and it appears when you choose something, or you have a target in your head and this flashes in front of you, so you're like, yeah, this is what I wanted. So this P300 appears, and it has been used a lot in brain-computer interfaces. So briefly, I think brain-computer interfacing is phenomenal, is a fascinating field of work, and it's basically using machine learning to have that loop of you acquire some EEG data, you process it, so signal processing, you extract some features, you classify whether you have found that brain response, and then you provide feedback, and all of that happens in real time. And in using that, some people have been able to spell, and some people have been able to control wheelchairs, not always with the P300, but it's very fascinating. I would love to talk about this more after. So tying this back to the paper that I want to discuss, you look at brain-computer interfacing in a lab setting where I'm just sitting here, just making sure that there are no electrical interference, and it's okay, you can make it work. But then you go into the ICU, and then you go into the surgery room, and the electrical interference by all the computers around you is insane. So this just speaks to how much work went into this project over here. So what the authors did is try to see if a person, after they have gone through coma, so you get into a coma after trauma, and after approximately 1 month, you emerge, and that you emerge as either completely healthy, which is great. You can then be less good by being in something called minimally conscious state, which you can kind of follow, but you're not fully there. And people don't really know what is happening during that period of time. And there is vegetative state, which is eyes open, but really nothing is happening in terms of behavioral responses. And then there is the locked-in syndrome, where typically happens when the patient has absolutely no control over their motion or their motor abilities, but they're fully there. And there is one book that is this pretty much the thoughts of a locked-in syndrome patient talking through blinking and writing a book. It's fascinating stuff, terrifying. So what these authors did is use some training by providing trials where they tell a patient whether to say no or yes. They tell them from beforehand. And these patients, they're healthy. Some of them are vegetative, some of them are minimal conscious, and some of them are locked in. So they tell them. Sometimes they don't know if they are actually hearing. So they tell them what to do, and they record for a particular piece of time. There are no responses at this point. And this is a 4-class classification. So it's no, yes, stop, and go. They could only be yes or no. And then the actual trials is where they ask them questions. So things like, is your name Mark, right? And then if the patient's name is Mark, then you expect when it says yes, then there is a P300 to that yes. When it's no, because the patient's name is actually Mark, then it's not no, so no P300, right? And this is kind of where they had the feedback loop of the BCI, which tries to classify what was their choice based on their brain signals. And as with actually most BCI applications, it wasn't deep learning, it wasn't anything too sophisticated, I would say. It's linear discriminant analysis, stepwise linear discriminant analysis. And I think that's absolutely fine, and it's actually good because you can actually investigate and interpret it even more. And the point is not to use sophisticated tools that you can't really tell what they're doing, but actually see the what they found is if you look at this is just an excerpt just looking at some patients. So healthy controls, you find like you see this red and you see this gray, so they see a P300 in the healthy controls. In the locked-in syndrome, you can see a bit of a separation here as well. So again, this is time, this is amplitude. P300, so P is positive, so red, there is a P300 here. And then they say that in the minimally conscious state, which again, this person doesn't really respond, they have also reported seeing a P300 of sorts, which is, if you see a P300, it means that they're processing. If they're processing, it means that there is some sort of consciousness going on. It might be waxing and waning, going away and coming back, but that is a clear indicator that there is something happening in there. So if you look at the results as a whole, I think you could look at it as kind of disappointing. Well, you can do brain-computer interfacing in healthy controls, HCY. Y is young. HCO, healthy control old, is a little bit worse, but you can still see it working. There's 100% and online and offline. But then once you get down to locked-in syndrome, minimally conscious state, and vegetative state, it becomes lower. But I do want to bring your attention to the fact that this is a 4-class classification. And that's part of biology. In order to understand something this intricate, it's not going to be as straightforward as you can tell with 100% accuracy whether somebody's doing something. But there is definitely something there, and the authors conclude that it is a tool that can be used and should be taken to this population to actually understand whether they can process things, because if they can process things, maybe you should apply different types of rehabilitation or maybe talk to them a bit more. So again, very, very broad topic, and I'm definitely looking forward to talks in the future from people that do this in their typical research.
Oh, there was, I guess, 2 questions as well for here. If you could go to the previous slide. The first one is, I guess, using the linear discriminant analysis is taking in, I guess, these graphs and trying to find the P30. Is that kind of the general kind of problem outline of this?
Correct. So the input in training is, well, and testing, is that you get the values over time, and the values over time are, let's say they're 1,000 milliseconds in length, and you try to predict whether there is a P300 there or not. And typically this is so much data that it just overwhelms the model. So what people end up doing is do very engineered feature extraction, where, let's say, you only look at data in the 300 range, or maybe you take the frequency features in that range. You could also, just because EEG, you have a lot of electrodes. So which electrode do you actually look at? And some people look at spatial filters, which I will actually be discussing later.
I see. I realized I said P30, which probably means a very different thing, but yeah, P300.
I know it's a thing. I don't know what it does. It's just,
I see.
yeah.
Another question was, I guess these signals, the red and the gray ones, are the gray ones kind of a baseline, and are the difference is statistically significant usually, or not necessarily?
Right. So just in terms of what they are, so the red would be, let's say we told the healthy control, and I'm just looking at this because this is a healthy control and it's easier to see. So if we tell them that they should pick yes, then that red is yes, is when they hear yes. The gray is when they hear something else. So if they hear no, then no P3. So that's what happens. Is that clear?
Yeah, I think that makes sense. Thanks.
Okay. And just in terms of statistical difference, in most cases there is a statistical difference between the two, but the best part about machine learning is that it doesn't always need to be statistically different, because again, this is just in single trial the noise is too much, and sometimes people don't even bother with testing for significance, which is not good practice. But sometimes people just depend on what the machine learning is telling them and look at the accuracy as an indicator of statistical significance.
Gotcha. Makes sense.
Okay. So the last paper that I'm going to be touching on is actually a paper that we published. So my group has published last year, the end of last year. And this was kind of the culmination of, I think, a 4-year study looking into the effects of concussion, also known as mild traumatic brain injury, on the brains of adolescent like teenagers between 10 and 17 years old. And these were patients that had a concussion. They were exhibiting some symptoms, and they came to us, and they got better over time. So overall, concussion, at one point it was thought to be transient. So people thought that you get concussed, you exhibit some symptoms, and over a few weeks it's gone and everybody's great. Unfortunately, that's not the case. People are starting to understand a little bit more about it, and that it doesn't necessarily go away in all the cases. Also, that even when it goes away in behavior, so even if cognitively I can do the crosswords really good and I can navigate around my place, my brain might not really be doing exactly the same as before. And that you can see these characteristics of brain changes in neurophysiology. Okay, so back again to EEG. So just again looking at ERPs, and this is again similar to what I was just talking about in the previous paper. This is another instance, but it's not really looking at within subject, but between subject. Different people, when they get concussed, they show different things. Okay, there are some commonalities between them, and which we want to look at, but even two controls will look very different. Okay, so let's say, and this is just an example, like if you have a control that looks like this, and I don't really need to describe what this is, it's just different sets of responses. But just by looking at it, if you look at the concussed, you're like, it's the same scale. This is more scrunched up. It's more diminished in amplitude compared to controls. Okay, so there's something there, and if you look into the earpiece themselves, okay, you can kind of tell what's happening. But then if I give you this other patient that walked into your clinic and you want to know whether they are concussed or not, you can't really tell. It's very difficult. It's a very high dimensional data set, and the subject variability overrides what you as a human could do. So yeah, small data set, numerous covariates, people get concussed in different ways, and the only way we can look at it or have looked at it is perception of data, because you need to look at it in a particular way and statistically analyze that. But then as a human expert, you don't see the full picture. So what we did is actually, instead of doing all sorts of feature engineering and try to force the data, the EEG data, into something that I perceive and what my PhD supervisor perceived and my other collaborators perceived as things related to concussion, I wanted to devise a machine learning structure that takes EEG as a whole, all of it, and try to reduce that and deal with it on its own, and give me what actually separates those two, like concussed versus controls. So doing that was done by just first reducing all sorts of parameters, because again, very, very large high dimensional data set, by using maps. So this is using convolutional neural nets in a very particular way, which grabs a particular map across the entire head. Of this response happens when the middle of the head exhibits a positive response, but everything else is kind of negative or close to zero. And all of that is learned on the fly. Use all the trials. So in typical paradigms, you average the trials to look at high signal to noise ratio. We just use the trials as they are, and then after training on the trial, if we look at one subject's data, which could be between like 400 to 800 trials, if they're classified as concussed or as controls in one way or another, we just apply a very simple threshold at 50% and then put them in that class as a subject as a whole. And this
quick question here. Is the input data, like, was it captured as image data, or is it, captured as maybe numerical data?
Great question. So the data is just numerical data. So if you look at, so in here, you can see the cursor, right?
Yep.
Excellent. So let's just look at the gray box over here. So 64 channels. So this is the data, numerical data, coming from each of the different electrodes on the scalp, and each of the rows is one trial. So from 0 milliseconds after they have heard a particular sound to, I believe it was, 1,000 milliseconds after they heard that sound. So again, numerical, and then across, different channels, time. Time is the Y. So all of this is processed, and where the concatenation or the convolutional layer runs on all the channels in order to reduce all the channels into one channel. And that is what forms the map.
Gotcha. Thanks.
Okay, so using this on a group of subjects that was 48, I believe, 48, between controls and concussed, we were able to tell the difference between them with 85% accuracy, with sensitivity and specificity like ranging around that. I believe one was 84, one was a bit around 86, which is quite good, especially that in this context, looking at things purely neurophysiologically, not with behavior. So this is all well and good, and you could look at this as a success because you can tell apart two classes and yay, we're done. But the problem is, just because of small data sets, of course you're bound to overfit your data, even your test data, because there is, you test multiple things and you test a few architectures, there is bound to be some knowledge leakage, and there's just no escaping it, unfortunately, unless you do studies in separate locations. So what we did is use something called Chap Values, and I do recommend you to check it out. It's one way of trying to interpret models that have already been trained. And what that does is it takes the model and feeds in the input and tries to see how does the input, if the input gets changed, how do the classes change? And in doing that, it kind of gives you a feature importance metric. And if you flow that back through the deep network, you can actually reflect that and transform it back on the head. And if you transform that back on the head, you can start to see things. So if you have the front here is the nose, these are the eyes, and this is kind of the 64 electrodes. X-axis is time, and the y-axis is the mean Chap value, which is again just feature importance. This transcends the view of me trying to sift through it by eye and trying to tell what is different in the noisy data. This tells me that this model was able to differentiate between the two classes because of this peak that was happening in the posterior region of the brain. Oh, and there is also a bit of an earlier response that was a little bit more frontal in the head. There were things to the side, so again, temporal regions, and all of these are different things that should then be investigated. Not using feature interpretation and model interpretation, because this is post hoc, but then they feed into new studies to actually understand this phenomenon a little bit more. So, yeah, so if there are no other questions, I just want to end on a note of just, if I wanted to communicate three things out of this talk, it's that the brain can inspire machine learning, and the other way is also equally feasible and equally good. Machine learning can revolutionize the study of the brain. Neuroscience is a vast field, and all the things I've discussed are just scratching the surface, and in what's to come, as speakers will speak to, as well as just new work that we don't even know about right now. And the last thing is, machine learning in neuroscience is not just about basic scientist discoveries and basic as just science for science's sake and understanding the brain, but it's also a very important catalyst to innovate in real life clinical applications, be that in trying to determine whether somebody is concussed and tell what should be done with them and how to get them better, to tell whether somebody is in locked-in syndrome or vegetative state and should get the medical attention to potentially improve them a bit. And I just want to say, thank you for being here and for listening, and I'm happy to take any further questions.
All right, thank you so much. These are very, very exciting use cases, really, very... I guess I'll save one question for later. There is a couple questions about the third kind of paper that you mentioned. The first one is, so you mentioned reducing parameters with learning mappings or something on one of the slides there. One question was, could you have used PCA to do the dimensionality reduction, or is it related to what you mentioned here?
Right. That's a great question, because nobody really knows. A lot of people do use PCA. Some people use ICA as well. The problem with PCA is that it applies different constraints that might not be the case in EEG. And with PCA, you can either reduce in channels or you can even reduce in time. So it sounds like a very versatile thing to use. But the problem is that I believe it's the linearity assumption just makes it less usable. And this is, on my end, unfortunately, just an empirical finding rather than one that is based in theory and actually understanding why that is the case. Great question.
Yeah, that's definitely something I was curious about too, but definitely, I guess the domain knowledge really comes into play here, right? Regarding whether PCA techniques could not work with the data potentially.
Mm-hmm.
So another question is, let me just read that. So for this one also, did you use domain adaptation to increase the performance? And yeah, I think that's one question.
What do you mean by domain adaptation? So do you mean as in making sure the differences between subjects don't seep into the model?
I'm going to ask them to clarify. I think what they meant is when you train on other types of models for perhaps other types of injuries, and then using kind of that knowledge to help inform how you built, this model for a specific type of injury.
I see. I think I'm just sensitive to hearing domain adaptation because it's something that I really wanted to look into, and we can discuss that after. But the main domain knowledge has been transferred in some aspect, but it wasn't to feature engineer. It was to design the architecture. So the paper right before that was also using EEG and also using pretty much the same paradigm that the patients went through. They were concussed. Everything was kind of the same, but they were a lower number of subjects. So single trial wasn't really possible there. And the knowledge of the different channels and that different channels are kind of redundant because there is a lot of smearing across different electrodes. So you need to reduce it to give a helping hand to the architecture of the deep learning. Well, this is not really deep. For the model to learn a little bit more, that was guided by domain knowledge, but it wasn't guided by a separate kind of brain injury per se. And this is just from the fact that different injuries look very different, and we're not really at a point where we can equate coma with minor traumatic brain injury. They're very different. Hopefully at one point we can have that knowledge transfer. And also because there isn't that much data that is there coming from the specific experiment. Because the problem with any data coming from biology and going into a specific target like that is that this data, if you change one thing about the experiment, the output becomes very different. And that might completely confuse your model. It's like taking the input from different fMRI machines. Even though they should do the same exact thing, but the output is very different, and you need to bend over backwards to actually make them fit.
I see. There was a clarification on that question. So I guess they were asking about the domain kind of transferability as a means of how do you deal with out of distribution data was their kind of main question, or are there performance metrics on generalizability of this model? I
see. So the only way we could speak to the generalizability in this case is just cross-validation generalizability. We assume that they have been, like, these patients have been sampled at random, and that they capture the variance in the entire population, both of which are incorrect assumptions. And the only way this can be pushed forward is to establish that this is not just random variance in the data. It's not just overfitting to the data, which we have established, But then the only way to generalize it on the population is to actually do further studies where we take this model or a slightly adapted model that is similar to it and then test it on completely unseen people from different areas using different equipment just to ensure that generalizability.
Gotcha. So there's several other questions around the topic of, because the data set is small, so could bootstrapping be of use, or other types of data augmentation techniques for these types of samples?
So very, very good question. So we tried to do bootstrapping and data augmentation, not in this one, not in this study, the study right before that, which used more feature engineering approaches, which made it a little easier to do that. Again, this is empirical. This is an empirical answer, just trying it on EEG data in this context, but it didn't really boost the performance in any way. And I think this is just because we haven't really passed the point of enough subjects to actually bootstrap. It's too little that the coverage of the feature space, especially given the dimensions, is just not enough. Definitely something that needs to be explored a lot more on.
Gotcha. And I guess one other question is, because there's so many, I guess, like you said, right? Like biology is noisy. Could it be, because I do think that it seems to have captured a lot of information from, I guess, like EEG, like it's like coverage wise, but are there other types of data that could be used in these models, like other types of physiological data? But I suppose that is very hard to gather in real time and just in general, because you need live subjects.
I think that's actually a very interesting question, because you can add all sorts of data. The problem is, I think the problem is the more data you capture, the more features you add, and it makes it even harder to get more patients, because they need to go through different additional hoops, right? So I don't know, because for example, a lot of people do EEG with fMRI like at the same time, which provides more data, and you could say that this data is definitely going to enable you to be a lot more accurate. But the things you need to do to get these two running are it's quite a feat. I believe that it should be an iterative process. It should be you look at one modality, you go deep into it, you go through the loops of basic statistical analysis, machine learning, and keep going back and forth until you make this concrete. Okay, you assume that there is no interdependence between the different modalities per se. It's like doing greedy feature selection. You find what you need from that particular modality, then you move to the next, or other people's are doing the next, and then you combine them. But just throwing them at each other from the beginning, I think, is difficult at this point.
Gotcha. Yeah, there's definitely a lot of limitations as well. Like it's not necessarily the most is better, and a lot of domain knowledge goes into even selecting these features and getting the clinical trials, or whatever the term is. Another question was about, I think it was the second paper with the again, the P300, if I'm saying that correctly this time.
No, that's correct.
So it was back about the question about statistical difference between, I guess, the red and the gray. And they were wondering, if there was no statistical difference, how could you, I guess, reliably say that they are kind of different responses?
So when it has to do with just general group level studies, statistical difference is definitely important at this stage. When you look at this and I say that it's not statistically different, typically what you do in EEG is you take the average of whatever this peak is, right? And you take that same average from the other condition, right? There is no feature extraction. I think I'm just talking to it from a strictly neurophysiology perspective. But then if you look at these two values, it might not be significant. But the machine learning application of and the feature extraction component of that machine learning, the features that get found or the features that get selected by the machine learning would be significant. It's just that as a whole, it wouldn't be.
Gotcha. I guess a follow-up question to that is just to clarify if for example, the training data, were they labeled, for example, the red is, like there were training data that that the red means this type of thing and the gray means this other type of thing, right?
Mm-hmm. So let's say in trial one, okay? So trial one would have exactly the same thing as below here, right? So you know that in block one, you tell them, yes is your target. Okay? So then you flash no, yes, stop, go. And then you look at that response. Let's just say it's one electrode, right? And then you take the response to yes. You already told them choose yes. And you assume that they have done that in their head. And you label it as yes, this is a choice. And you look at the response to no, stop, go, and you label them as no.
I see. Yeah. So it's definitely based on the instructions as well. So that's actually a really fascinating point about like gathering clinical kind of data as well, because oftentimes, actually in other industries too, then some assumptions it really depends on what they do. They could click a button that said, yes, I have read this and I agree, but they might not have. But that's just what happens in all sort of things.
There are some horror stories, trust me. Like there would be like this entire like person set up with EEG on their head. They're going through things, and just the experimenter is not monitoring their responses, and they're just answering randomly, and that's it. So you don't even know if the healthy control or the patient, maybe they if they have hearing problems, you're done. Like you are literally adding incorrect labels into your data.
I see. Okay, one last question. Did the visualization of the feature maps, so in one of them that you mentioned, the feature maps inside the network help you hypothesize on how the brain works for that use case?
Mm-hmm. So I just want to confirm it's this one.
Let me see. So one that you specifically mentioned, feature like maps or some sort of reduction, or maybe it is this one actually.
Yeah, I think that's it. If just interrupt me if it isn't that, and they say that it's another slide. So the way I believe this would influence my understanding of brain injury isn't necessarily how the brain works. It's more of a tiered layer. It tells me that this particular response is affected after concussion. So then I look at that response and I try to say, okay, well, what is this response known to be influenced by, or why? What does it correlate to on a brain function overall? And then I make some assumptions and some speculation into, well, if that is the case, then this would mean that their behavioral function may be affected in this particular case. Or maybe because I know that this component is produced in this part of the brain, then maybe this part of the brain was injured in some capacity. It's also very difficult because these are functional. This is the functional side of the brain, so you can't really poke at it too easily.
Gotcha. I think that makes sense. So thank you a lot, Robert. Like this was very, very fascinating and actually a very good point on how neuroscience, kind of came round like a circle, right? How machine learning was taking inspiration from kind of the structure and of the neurons, and it's very, very interesting to be reminded of that, because I think right now when we say neural network, sometimes I'm not reminded so much about where it came from, to be frank. But I'm very, very excited for the stream, and thank you so much for today.
Yeah, thank you very much. It's a fascinating field, and I really am looking forward to the next few talks. Thank you very much for facilitating, and thank you for everybody that's here.
All right, thank you. Have a good
Have a
day,
great
and thanks
day.
for tuning in.